אז צהריים טובים לכולם, ותודה שאתם משתתפים בוובינר שאנחנו עורכים היום, תחת התוכנית BioConvergence שרשות החדשנות מקדמת, ובמסגרת התוכנית אנחנו שמחים על שיתוף הפעולה עם מכון פראונהופר, ובספציפית עם מכוני המחקר בתחום ה-Life Science שלהם, שמשתתפים איתנו היום בוובינר. So hi everybody from Fraunhofer Institute. We're really so excited that you are here with us, and we can share our same insights together today and our views with the audience. I would like to call Anya Eldan, VP in the Israel Innovation Authority and heading the BioConvergence National Program, to say a few words about the program and this kind of a collaboration that we are promoting today. Anya, please.
That's not gonna work. Hello. Now I'm not mute. Thank you very much, all the guests, and thank you all very much, all the participants. We're very excited about this because we've been promoting bioconvergence strategy for over a year now. And you will see in a minute that we really need partnerships. So we're so very happy to be able to consider partnership with Fraunhofer on this front as well. We have a lot of other collaborations. So a few words about Israel Innovation Authority. We're a government agency. We spend about €500 million a year supporting Israeli innovation. Our job is economic, so we use innovation and we use our money to promote the economic prosperity of the country. So we want to make our companies as competitive globally as possible. Oh, and now I have a slide. And you'll see we do it through a number of ways. We strengthen the innovation ecosystem, we enhance economic impact, but one of the most important pieces is we enable emerging disruptive technologies. And while Israel is very well known for being a very successful ICT, information technology economy, about 1/3 of our money is spent on life sciences. So both pharma, digital health, and medical devices. And we have established, after long research, that bioconvergence, that convergence of biology and engineering, is going to impact healthcare in a very major way. Not just healthcare, it's going to impact food tech and agri-tech and energy, but healthcare is what we want to talk about today. And the Israel Innovation Authority has decided to lead the bioconvergence strategy. We have implemented investment tools that we provide. We have implemented a program, national program that's about to kick off, that's going to include other ministries and academy. And most important, we also implement international collaborations. So for Israel, this is a growth engine for us. So for Israel to be one of the leading clusters in bioconvergence, we understand that we need collaboration, we understand that we need partnerships, and we're reaching out to the most important research organizations worldwide. And it's not a coincidence that one of the first, maybe the first that we reached out is you guys. So it's great to have you here, and I'm sure this is going to be a very interesting event. It's going to be a beginning of a collaboration on bringing the bioconvergence, the technology, to affect the healthcare. So thank you very much.
Thank you so much, Anya. I'm welcoming Dr. Itai Kela, or science heading, the science manager of this bioconvergence program, to say a few words about the program. Thank you. Please, Itai. Itai, you are on mute. I'm sorry, one cannot hear you.
So again, I'm happy to be here. Thank you very much, and I will try to take you very briefly about this new term, the bioconvergence, and what are the Israeli strengths in this new field, and what we are starting to do in Israel in the last year or so. So we are defining bioconvergence as a merging of tech with life sciences. And you see this graph, and it's basically integrating intersection of data, computing, engineering with life sciences, with genetics. And we see today, and I give you show you briefly one slide about the market. We are today in the tipping point where the industry is urgently needs this kind of new innovation engine that will drive the health tech sector, and as Anya mentioned, also will drive other industries. To simplify it, we see the bioconvergence, or we call it engineering biology, that biology is in the center, and basically a hinge of integrating other disciplines to solve unmet needs and challenges also in healthcare and other sectors. So recently, McKinsey published a report about this field. They call it biorevolution, but basically the same, different terms to the same field, and basically say that bioconvergence mainly focused on healthcare, but about 50% of the applications will transform, will be part of other industries like agriculture, materials, energy, consumer products, defense, and others. And this market is huge. It's $4 trillion a year for the next 20 years. And you see the numbers, about 30% of the R&D spent will go to bio-related products and R&D. And so this global trend, the bioconvergence, is not specific to Israel. It's a global emerging industry that is just starting to transform the health tech sector. We can define the three main impacts that bioconvergence will drive. The first one is to engineering the drug development, or engineering the whole process, all the traditional process of development by robotics, automation, using data, AI, and other disciplines. The second one is advanced therapy, gene, cell therapy, CRISPR. All these solutions, therapeutic solutions, are going to be the main products in the next 10, 20 years. And the third one is designing nature, meaning using genetic tools to engineer biological systems, also for healthcare, but for other applications. There are many applications also in, as I said, in agriculture and also in defense. So these are the main three impact factor that bioconvergence will drive. We see just an example. What we mapped is that are the main fields, and also have some activity or major activity in Israel academia, and starting to penetrate to the industry. Talking about therapeutic discovery, about you see the lab on chip. Talk about advanced delivery systems using biorobotics, cells, viruses, engineered systems to deliver more specifically drugs to target in the body, talking about advanced therapy, talking about 3D printing, tissue engineering. All of these fields are now the main, maybe I can say the main drivers of this wave of technology. Other fields like bioelectronics, engineering smart materials, optogenetics. So that's just an examples of what we can relate to bioconvergence. And as Anya mentioned, we in the last year or two have developed a national program. That's the main rationale is designed or based on Israeli strengths in engineering and AI and the life science that we have in Israel, in academic in Israel. And the idea is to integrate these disciplines to develop a new cluster in Israel. And the vision is that in the next 10, 15 years, Israel will develop a leading cluster, a bioconvergence clusters with significant companies and a dozen of small, medium-sized companies that will drive this ecosystem, and will have a crosstalk with the academia, with medical centers to generate a highly integrated ecosystem. So very briefly, the program is focused to have two main legs. One is focused on science and technology, in designing new multidisciplinary education programs, increase the multidisciplinary research grants, developing research centers, increase the deal flow from the academia to industry by designing and improving the whole translational process, develop a new workforce that have skills both in life science and engineering, and to leverage this activity to develop a new Israeli new companies, to attract foreign companies to invest in R&D, in multidisciplinary R&D in Israel, and to invest and develop a new manufacturing program. Also, very importantly, we put a lot of emphasis on global collaboration. That's why we are meeting, and we understand that Israel needs collaborators, needs help to leverage and to promote this industry to the global level. In the last year, we started we have a lot of activity in the academia, how to mature research projects into companies. We have some calls for applied research. We also have some activities supporting companies, bioconvergence companies, innovation labs that we just attended, we just launched recently. And also, as I mentioned, we have a very intense global activity in several countries, in bilateral calls and other activity. Maybe I'll stop here. This is just an example of what we are doing. Consortia in genomic engineering, as I mentioned, AI Pharma Innovation Lab, bioconvergence calls in academia, human capital training programs. We also have a close collaboration with the Ministry of Health to design an innovative regulatory framework to make it easier for Israeli companies and foreign companies to submit, to enter to first in human in Israeli medical systems, and also global collaborations. So thank you very much, and I'm looking forward to this event. And Karina, your call.
Thank you so much, Itai. And I would like to, I'm very excited to have a warm welcome to our Fraunhofer partners here. And I'm calling Dr. Klaus Dieter Kugel, who is the head of the Central Office of Fraunhofer Group for Life Sciences, to present himself and his colleagues as well. So Klaus, please, Dr. Klaus, the stage is yours. And please unmute yourself. Klaus, you are on mute. Klaus, you are on mute.
My name is Klaus Krockel. Can you hear me now?
Yes, please.
I'm the head of the Fraunhofer Group for Life, from the central office of the Fraunhofer Group for Life Sciences. May I welcome you all very warmly. I would especially like to thank Mrs. Karina Rubinstein, who organized the seminar so excellently. 4 scientists are taking part. 3 from the Fraunhofer Group for Life Sciences, 1 scientist from the Fraunhofer Group for Production. So I introduce to you Mr. Andreas Taube. He is working for the Fraunhofer Institute for Manufacturing, Engineering and Automation, IPA, in Stuttgart. He's from the Fraunhofer Group for Production. I introduce to you Professor Steffen Rupp from the Fraunhofer Institute of Interfacial Engineering and Biotechnology. He is based in Stuttgart. He has also a project center together with the Hebrew University. He will explain to you about that later. I introduce to you Professor Friedemann Horn from the Fraunhofer Institute of Cell Therapy and Immunology. This is based in Leipzig in former East Germany. And I introduce to you Dr. Kamran Hornejad from the Fraunhofer Institute of Toxicology and Experimental Medicine. He's in the branch which is based in Regensburg for personalized tumor therapies. Can you see my presentation?
Yes. If you can put it on view mode.
Okay, I hope you can see it now.
It's okay, we can see it.
Okay, so the agenda of my talk is, I explain to you about the Fraunhofer Gesellschaft. You see already?
Yes.
Now you see the agenda, yes?
Yes.
So the agenda is, I explained to you about the Fraunhofer Gesellschaft, the Fraunhofer Group for Life Sciences, but especially also about innovation principles to build a bridge to your idea of bioconvergence. The name patron of Fraunhofer is Josef von Fraunhofer. He was a researcher, inventor, and also an entrepreneur. And he is this type of personality, which is our ideal personality within Fraunhofer, because he was able to bring a vita contemplativa, so to go in an introvert stage, to work at the desk, to walk outside, to create, to find new ideas, together with a vita activa, to go to the market, to explain to other colleagues what he would like to do. And to bring this together is very difficult in life. Sees both sides. Fraunhofer has now a staff about 20,000 people, 72 institutes, and more than 70% from our budget is derived from contracts with industry and from publicly financed research projects. So we are doing also contract research for the industry in Israel, and we are trying now to organize a funding with IIA for joint projects, because 70% of our budget we have to earn. We are now worldwide active. We also have a center at the Hebrew University, and later Professor Rupp will talk about that. We are formed in groups. This has a great advantage that we can easily push forward new innovations. We have the life science group, microelectronic production. Then we make meetings and seminars between these groups. And then the medical doctors, the biologists, the chemists talk about the problems they have and the solutions they would need to the engineers. And finally, our engineers are finding these ideas. So within Fraunhofer, we can work very, very interdisciplinary. We created here projects, which was a project which was called Automated Tissue Engineering on Demand, bringing biology together with production technologies, or RNA based biomarkers. Later on, Professor Horn will also explain to you about these types of projects. The background is a little bit also the Chicago School of Philosophy and Sociology, George Herbert Mead, he's always in my mind when I organize those seminars, because his specific teaching was that we have always success in learning from each other and with each other when we are able to take the role of the other. So when the engineer understands the biologist and the medical doctor, what he actually needs, and this is a unique position of the Fraunhofer Gesellschaft to be able to make this type of interdisciplinary research possible. And we have to find a language that the scientists, that the engineers can understand the life science scientists and the medical doctors. We are very active in patents, of course, and in licensing technologies. Our great success here is also the MP3 success, which is famous worldwide. Within the Fraunhofer Group for Life Sciences, we establish now 4 business areas: health, food, bioeconomics, and environment. We were established in the year 2000, have 6 member institutes. 3 of these member institutes are taking part today. And in the field of bioconvergence, we have, of course, we work also with this idea of bioconvergence. For example, the Institute of Toxicology and Experimental Medicine, they told me we use multi-omic molecular and functional data for early cancer detections, monitoring and individualized cancer therapy, as well as in preclinical biomarker and drug development R&D. And later on, Kamran Hornechat will give you also deeper explanations about his research. Similar approaches we have by the IGB or in the production group. The Fraunhofer Group for Life Sciences started with 4 institutes. Now we are all over Germany, very active.
Klaus, I'm sorry, but would you like to flip the presentation slides, because we are still on the second slide? Would you like to show other slides?
I'm from my side, it is now?
Now we are on the fourth slide now.
Now you're on the 4th slide. Pardon. I thought this was a slide. So this is life sciences are now all over Germany. So I have here different screens. Pardon. Thank you for giving me this information, Karina. The changes in the next 10 to 20 years. So I think from the Fraunhofer Foresight report, we have a long-term relevance of artificial intelligence, liquid biopsy, microbiome, biodiversity, and bioeconomy or wide industrial biotechnology is only possible when we have also circular economy. So you see, these are all somehow life sciences disciplines. I explained to you about the 4 business areas because we are concerned health today. I only say about food. Okay, prevention of diseases is very important. And so we always have to consider also about food developments to realize that. In the health area, we work nowadays with a 4D concept with drugs, diagnostic devices, and data, because within Fraunhofer, we can bring these 4 areas together with our multidisciplinary approaches and create their synergies. We have licensable products and processes like cell, gene, and viral therapies, in vivo test systems. And of course, we offer contract research to you, research and development, therapy development for immune-mediated diseases, cell and gene therapies, precision medicine, animal models, in vitro tests. We are active in these areas. We are concerned about the environment and bioeconomy. You know, we have this pandemic diseases, which also will be a greater problem in the future because we are destroying nature, and the biodiversity cannot protect us in the ways like before. So we are very concerned about environmental sciences too. That's again our 4D concepts, where we bring together data, diagnostics, devices, and drugs. With the different technologies we have in our 6 institutes and the research institution. I think in the past we thought data is important in diagnostics, drugs, and devices. But I by myself, I think if I go in control theory by general negative feedback control system, the data will be the leading role in the 4D. And we all have to consider that very early. And I think Israelis, they are doing a very good job to bring life sciences together with IT on a very high level. I like metaphoric thinking. So in Germany, when we speak about life sciences, we speak about the color spectrum. Agrobiotech is a green biotechnology, marine biotechnology is a blue biotech, industrial biotechnology is a white, medical biotechnology is a red, or the yellow one is the insect biotechnology. And of course, if you switch or change the track from one color to the other color, so it's the underground railway system here of London, then you can go to wide to faster destinations. You have more interdisciplinary approaches. So scientists have to be very open-minded to realize that. And you have to bring the color spectrum together and to make your scientists very interdisciplinary. And we are developing those approaches very much in the Fraunhofer Group for Life Sciences together with the other groups. For example, we started in the green biotechnology, thought about the Caucasian dandelion, how to change the Caucasian dandelion with breeding research and genetic engineering into natural rubber. And we were successful, and now we have wonderful contracts with the car, truck, and bicycle industry for tires, and a lot of demand from the shoe and mattress manufacturers who also want to have our natural rubber. So we changed here the track from the green biotechnology to the white biotechnology. So we have to think about biodiversity, the genetic information which is exchanged there, the data interpretation, and we have to create AI, artificial intelligence, and self-learning systems together with a color palette of biotechnology. That's my philosophy in the background by our innovation projects. We do by Fraunhofer the applied research. It's our task to finally reach industrial implementation. And that's also the reason why we are coming together today. We are doing in general contract research, and we are also looking for funding programs to do international projects. That's the reason we are working today together with the IIA. Yeah, that's an example I already mentioned, that we develop several different cooperation models for strategic partnerships and joint research. And here in Israel with the Hebrew University in the Fraunhofer Project Center for Drug Discovery and Delivery. And I think in the future, we have to consider very much how we evaluate projects. These are the sustainability goals of the UNESCO. That's a very project which until now brought us only some revenues. But if you see how many goals of the UNESCO are fulfilled with this project, like zero hunger, good health, affordable and clean energy, and so on, then you see that's the real value of this project of sustained exploitation of fish resources in Malawi by our integrated agriculture, aquaculture system has a very high value for mankind. And of course, we have to bring mankind together to create a better science. Yeah, these are my ideas, Karina, dear audience.
Thank you so much, Klaus, for this really insightful presentation and for sharing your philosophy. I think it collides with our philosophy by all means. Specifically, when we're talking about bioconvergence and the multidisciplinary way of thinking, doing science, the collaboration between engineers and scientists, I think it's really resonating to our thinking and vision as well. We can see a lot of ways that we can promote collaboration here. I think specifically in this event, we would like to promote research that was already ready for commercialization from your side in the areas of bioconvergence. And on that note, I would like to welcome Dr. Aviv Ze'evi to say a few words about how can we help and fund this kind of a collaboration. Aviv, please. Aviv, אתה על מיוט.
Okay, can you see the presentation?
Yes.
Okay, so
you can put it on a viewer mode and not on a presenter mode.
Sorry, I'm trying to display setting, and now it's okay?
Yes.
Okay. So hello everyone. So my name is Aviv, and I am a colleague of Anya. I'm responsible for the technological infrastructure division, and as though I'm responsible for collaboration between academic partner and industrial partner and tech transfer. And I think one of the main funding tools that we are talking about is this tech program tool, and I will try to explain about it. So this specific tool has three main goals. The first one is validation of academic or laboratory results. It means that the company has to make sure that the knowledge that's to be transferred to the company is works fine also in reality, I would say. The other one is adaptation of the technology development by the research institute to the industrial cooperation. It means that for sure the specific knowledge was developed according to the researcher assumptions, and some of those assumptions are a bit different in the specific company which is interested in the knowledge. And the third one is the development of the relevant skills, which means that the company will be able to keep on developing the knowledge by itself without the need to be completely independent on the research institute. So the main idea is actually to close the gap between the knowledge as it was developed by the research institute to the needs of the Israeli company, which is interested in this specific. So who can apply? Of course, academic research groups or research institution. It's this specific model is open both to Israeli academic research groups and to foreign ones. In this case, we are talking, of course, on a German research institute in most cases, and we have funded in the past 12 different projects with Fraunhofer participation. Also, 3 of them only this year, and it means that we have some good example of this kind of cooperation. Of course, we are looking for the Israeli industrial cooperation, which wishes to absorb the innovative technologies. It means that an Israeli researcher from the Israeli academia can join as well to this kind of collaboration if it's really needed and relevant. It means that in some cases, the knowledge is even more relevant when there is this kind of collaboration, and we will welcome this kind of collaboration as well. So what do you get? The company who submit the proposal will get 50 to 60% of the budget. The duration is up to 24 months and up to 3.4 million shekels, which means more or less 850,000 euros. This specific budget is allocated both to the company and to the researchers. It means that they should use it. Both of them should use it. And of course, the research institute might get directly, will get directly the funding from the Israeli company, and he will be a subcontractor of the Israeli company. Before I go to a specific success story, I would say that we foreseen a collaboration in which the Israeli company wants to commercialize actually the knowledge that was developed by the Fraunhofer Institute, and it can see the potential, the economic potential. It means that we will follow the project in a way to see if there is a commercialization of the knowledge in any of the products that will be developed post the project. It means that we will not fund any development of a new IP, which was created by Fraunhofer, but we are looking for an existing IP, but we will fund any adaptation of the knowledge to the needs of the company, and of course, skills, teaching the skills or instruction, the relevant skills to the Israeli company employees. And of course, it might lead to a further collaboration between both of them. And this can happen in different funding tools we have for product development, but not by this specific instrument. So I would like to show with you one specific success story. It's an Israeli company. Hill applied. It started as in the incubators program, which Anya is leading. And later on, they started developing advanced system for treatment of radiation against cancer. And the knowledge that was really needed for them was a magnet design for a proton accelerator. And they have identified this specific knowledge in CERN in Switzerland. And the achievement that they were able to get was half of the area required for the original system, a quarter of the volume, a third of the price, and 1,000 times lower weight, which means that the development of the specific magnet was taking them to a new ability and new option that were not there here in Israel, and therefore we needed this knowledge which was exist elsewhere. So we are hoping to find the specific knowledge that do not exist here in Israel, we hope that the relevant knowledge will get transferred to the Israeli company, will be one that gives her the specific market share. So we are not really looking on a knowledge that will focus the Israeli company only on the Israeli market, but on really to be in the as a world leader in this area. So as one of the division which is implement these specific tools, we hope to get a list of technology and knowledge that you are socializing, and we hope that to see that there is some kind of a standard agreement for the transfer of the knowledge that Fraunhofer can supply us, some kind of a statement that there is no legal problem of the transfer knowledge to another country. And of course, we hope to get an exclusivity of for the knowledge, but this is not mandatory. That's all. Thank you. If you have any question or anything, I will be glad to answer. I don't know, Karina, if we have time.
Questions will be answered at the end. We are receiving questions as it is through the Q&A, so I'm trying to respond as much as possible.
Okay.
Thank you, Aviv, so much for that. I think if there are any specific questions on the funding mechanism and so on, we'll be happy to reply to that. And also after the webinar is over, you're welcome to approach us through mail or by phone and so on. But I would like to now present the researchers from the Fraunhofer Institute that will present and talk about, describe their research specific areas which are more relevant to bioconvergence. And the first speaker, I'm welcoming Professor Dr. Stefan Rupp from Fraunhofer Institute for Interfacial Engineering and Biotechnology, IGB, Deputy Director and Head of Business Area Health. Dr. Stefan Rupp, please.
Oh, thank you very much. I'm just trying to share my screen. Let's see. Let's try again. Okay. Do you see my screen now?
Yes, if you can put it on a viewer's mode.
Okay.
Thank you so much.
This should do it. Okay. Thank you very much for your introduction. And I would like to give you in the next 10 minutes a very brief overview on what we are doing at the Fraunhofer IGB in the area of precision medicine. To give you a very brief introduction into the institute, we are located in Stuttgart in the southwest of Germany. Our institute has about 340 employees, and we have an operational budget between 26 and 27 million euros per year. Fraunhofer IGB is active in 3 business areas: health, sustainable chemistry, and environment. And I will focus today only on health. To give you an idea what our intention is on smart biomedical engineering for precision medicine, here a very brief introduction on the new paradigms in healthcare industry. As you all know, big pharma is moving away from small molecules that used to be a population-based medicine, heavily relying on animal testing, towards more precise medication like biologicals, theranostic approaches, and cell and gene therapy. And to do that, you need to focus on individual aspects, especially immune system components of the patient. That is basically precision therapy. The animal testing tools we have available, which are still heavily used, are not fit to provide exact preclinical data and that. So human-based in vitro systems are urgently needed to fill in a gap here. And one of our ideas is to supply this need for new physiological human test systems. To do that, we have a cell and tissue technology department headed by Angeborg Kendischer and Peter Loskill. And they are developing screening and test systems for precision therapeutics, which can be offered as a contract basis. We do have contracts with big companies like Novartis or Boehringer Ingelheim on that. Principles here, organ-on-a-chip models are made in-house, including the tailored microfluidic platforms. So they are produced in-house, and then you can integrate different type of tissues, heart, adipose, retina, into these organ-on-a-chip models. We have several disease models I will go into in a minute, and immune integration is a critical part here. On the other hand, we have a cell and tissue engineering department, which is focusing on genetic manipulation of primary cells, of cell lines to create reporter systems, special cell-based assays that operate according to GLP. And these are also integrated in 3D structures, 3D epithelial models that can mimic disease models, including components of the immune systems. For example, we have a toll-like receptor skin, reporter skin, which is also IP'd, so patents are available for this. To give you a very brief overview of what we have done in this area of organ-on-a-chip, here you see a kind of a toolbox of different systems we have developed: heart on a chip, retina on a chip, pancreas, and so on. Several of them carry IP, have patents on it. And I want to point out one thing: for several of the parameters you want to look at, you have an online monitoring. Here, for example, you see a calcium flux in a heart tissue. The same applies to 3D tissue models. Here we also have a kind of a toolbox system where you can combine different matrices containing fibroblasts and immune cells with different epithelia. So you can build up skin or oral models, for example. You can integrate reporter cells, immortalized primary reporter cells, for example, on toll-like receptor activity, immune receptor activity, stress response. And you can build up different type of disease models with it. You can look at immune response and modulation, host pathogen interaction, and importantly also drug mode of action or cosmetic mode of action. The matrices we use to build up these systems, be it organ-on-a-chip or the 3D tissue models, are also made in-house in the Department of Functional Surfaces and Materials, headed by Michaela Müller. And here we are able to modify biomolecules like gelatin or collagen, hyaluronic acid, and so on, to provide kind of bioinks with defined viscosity and also defined milieu for different cell types. These are then printed in the respective devices and can be used within the various systems, organ-on-chip or 3D tissue models. The second big part we have at Fraunhofer IGB is the group in vitro diagnostics. Kai Sohn is heading this group. This is basically a next generation sequencing facility, which can of course be used to look at the tissue models I just introduced you in more detail. The main IP here is on data-driven diagnostics. So basically, Kai and his group are sequencing biopsies of different types and provide a clear diagnostic readout. To give you a more detailed view here, what our expertise is here is from bedside to the ready probe going into the sequencer, how to prepare these biopsy material for sequencing, which biopsies to take for which indication, and then also provide the bioinformatics to read out a clear diagnostic profile for the physician. We have spun out a company on that, Nocendo, and this company is also involved in a large prospective non-interventional multicentric study called NextGenSys, which is currently running to get this method into the market in a certified way. Bacteremia and sepsis is the main goal here. This know-how is of course translated to other indications, different kind of bloodstream infections, infectious endocarditis or CNS infections. And recently we also have started highest throughput screening approaches on SARS-CoV-2. This is also together with other institutes of the Fraunhofer group. Friedemann Horn certainly will also be talking about that later on, I guess. Okay, the third group is headed by Susanne Beiler. I think she's in the audience and may be available also for questions later. It's on virus-based technologies, and here we try to design viruses and phages for medical or biotechnological application. The main tool is here herpes simplex virus. The goal here is to create oncolytic viruses, a combined virus immunotherapy. And here the genetic engineering of the virus is done in-house, the Fraunhofer IGB, providing it with a targeting moiety and an immune modulator component. We do have, of course, the in vitro models, 3D tumor models in-house and in cooperation with other institutes. You'll see here on the side several institutes you will hear today, for example, the ITEM and ITSI are part of this large program. And in the end, we are also able to produce substantial amount of a virus at a GMP level for testing even into clinical phases. The second part here is the use of phages, bacteriophages, genetically modified phages that can be used as an alternative to antibiotics, targeting directly pathogenic bacteria, but can also be used in a non-medical area like control of biofilms in the environment, crop protection, or food preservation, for example. Last but not least, I would like to introduce the Fraunhofer Project Center for Drug Discovery and Delivery at Hebrew University. I have here a collaboration with Professor Gershon Golomb and Amiram Goldblum since almost 10 years now. Our focus is on drug discovery and validation in combination with drug transport and release. And we use our 3D tissue models you have seen, organ-on-a-chip models, in combination with new types of anti-infectives and TLR modulators to find new therapeutics. We have, for example, several patents. Here, one joint patent is on TLR9 antagonists, which is also something we would like to commercialize. So with this, I think my 10 minutes are already over. I hope I could give you a brief impression on what we are doing. For most of the things I showed you, we do have IP, existing IP, or several technologies are in the patent process. And there are, I think, many options to start collaborations on existing IP. But as I told you, we also are a service provider for all the technologies I've shown you, which could be of interest to some of the companies here. Thank you very much. And here's the people who do the actual work of the departments.
Thank you so much, Professor Dr. Stefan Roh. Klaus, maybe we're gonna put on the screen at the end the contact information of the people in Fraunhofer that could provide more details on sorts of collaboration and the IP which is relevant to this event. Maybe we can put it at the end of the presentations of this webinar. So I would like to present now the next speaker, Professor Dr. Friedman Horn, who is from the Department of Diagnostic, Revolution Biomarker Center, Innovative Biomarker Development and Corona Field Study. He's the head of the Department of Diagnostics and Revolution Biomarker Center, Fraunhofer Institute of Cell Therapy and Immunology, IZI, Department of Diagnostic. Professor Dr. Friedman Horn, please.
Yeah, hello together, and thank you very much for the invitation. I'm very humbled to be able to present our groups and my department here. I would like to
maybe
share my screen.
Dr. Stefan can just stop his sharing.
Exactly.
So you'll be able to share your screen.
Mm-hmm. Yeah.
Let me see. Okay.
Right.
I guess now you can go ahead.
All right, thank you very much. I can show it. I hope it can be seen. Is it visible?
Yes.
Yes.
Great, thank you very much. Yeah, thanks again for the introduction. And yeah, I would like to present here shortly the Fraunhofer IZI as an institute, which is, as you see, located in the former East German part, a little south of Berlin, in Leipzig. The institute actually was founded around 2005, so it's 15 years old, and its main focus actually is on, as its name says, cell therapy development and immunology. I show you very shortly the departments of this institute. We do have two main departments that are involved in developing and producing cell therapeutics, in particular for CAR T cells. Actually, our GMP unit here is Europe's largest production plant for cell therapeutics for the CAR T cell area. And one of the products which we, in collaboration with Novartis, produce is this Kymria cell therapy product for treating leukemias. So cell therapy and its development is a major focus of the house. However, we have also other areas of research. One is therapy validation, also a GLP test facility, which we can use to validate and to monitor therapies, to do safety studies and so on. We have a department of immunology, which is very strongly involved in developing vaccine technologies. Actually, originally for example, for the West Nile virus and viruses of this class. But now, of course, with Corona coming up, we have also a strong focus on developing vaccines in that area and developing also novel technologies and strategies actually in this context. Then there is a department of cell therapy, which also is involved in cell therapeutic and vector therapeutic approaches, and also contains a large experimental imaging unit. And finally, my own department of diagnostics, where we have a multitude actually of approaches which are brought about by 6 different research groups in this department. There's a group of microdiagnostics of Dr. Kuhlmeier. They are doing lab-on-the-chip and point-of-care developments for diagnostic devices in the infectious disease field, also in oncology and other areas. We have Dr. David Smith with a group on DNA nanosystems, so very smart and innovative approaches to use actually DNA and other nano devices to do either diagnostics or even therapeutic approaches. We have a inflammation model and immune diagnostics group headed by Dr. Lange. We have a strong group focused on bioinformatics, particularly on RNA bioinformatics, because one of the major foci here, which we are actually concentrating on, is also RNA next generation sequencing, which is conducted in the research group of Dr. Blumert, next generation diagnostics group, in order to raise and discover and validate novel RNA biomarkers, which I in a minute come to. And finally, we have the group for cardiomics of Professor Oberbach, which actually is analyzing, for example, infectious pathogens for endocarditis and other heart-related diseases, as well as doing research on other areas of cardiac diseases in a very innovative manner. Within the department, we have established a biomarker center, so-called Rivolution Biomarker Center, where Rivolution stands for ribonucleic acid derived diagnostic solutions, which actually also already expresses that one major focus of this Rivolution Biomarker Center is actually about RNA as biomarkers, in particular also RNA biomarkers from the non-coding RNA field, which turned out to be the vast majority actually of RNA molecules in the cell. And what we do here, we have established actually a whole value chain reaching from the clinic, from the clinical samples through RNA biomarker discovery by next generation sequencing to validation qualifying RNA biomarkers for certain diagnostic tests, finally transforming them into analysis algorithms, and finally helping the industry to register that after validating these biomarkers by clinical studies. The entire process actually is established here. We are certified by ISO 9001 for it, and have actually put together all the know-how and technology which is required to cover this entire process. For the shortage of time, I cannot really show you results of that, but I would like to shortly mention that we, for example, have by this value chain already established very interesting proof of concepts for diagnostic tests, for example, for prostate cancer. Prostate cancer tests from urine, which are much more sensitive than the commercially available ones, which we now try to, in collaboration with diagnostic companies, to commercialize in the future. We also have a set of biomarkers, or actually a signature of biomarkers, a very large one, identified, which can also predict the prognostic behavior of prostate cancers, and so by that mean help decide the physician on which therapy to use optimally for a particular patient. So these are just two examples actually, but as you might imagine, we also look at a number of other diseases like chronic inflammatory diseases or other tumor diseases and so forth with our value chain and with our biomarker center. Finally, I would like to present here a very new study, actually, which we just are in the process of starting. Saxo-CoV. So what we are doing here is we have in collaboration with and by funding of the Ministry of Research in Dresden for the Free State of Saxony, we were able to start a very large study on the corona pandemics in Saxony. The consortium is a very large one. We have involved a number of universities here from Leipzig, from Dresden, also from Poland, Wrocław. We have Fraunhofer IZI, of course, as a coordinating institution. We have involved the Helmholtz Center for Environmental Research in Leipzig, the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, several hospitals, and also official, administrative, and health institutions. So a very large consortium. And what we do here and our aims actually are actually to find out what actually, the principles of the pandemics here, particularly in Saxony, are. If you look at the current numbers, you can see that in Germany, actually now the current status of novel infections every day actually already surpasses the first wave in March and April. And the same thing on the left is true on the lower scale, of course, in Saxony. But here also we have much stronger and much higher values already of infections daily, on the daily basis. And if you look around to the neighboring countries for Saxony, particularly Poland and Czech Republic, actually the picture is, I would say, almost horrifying, because the numbers just go beyond but go up in an unbelievable manner. So what we try with our consortium here in our study is we try to collect really comprehensive data on very, very many aspects of the pandemics. We look into the prevalence of infections themselves. We look into the immunity and antibody, seropositivity, one should say, of people. We look into the genomics of the SARS-CoV-2 virus and its development, but also using it as a tracking tool actually for tracking the pathways actually that the infection goes between people. We are looking into infection distribution patterns by helping the official sites to track the infection in the Free State of Saxony. We do surveys on contact behavior, on symptoms. We look into the COVID-19 disease parameters from the immunological and clinical standpoints, and many more. So it's just actually examples, but the collecting these data actually, we hope will help us to get a real comprehensive picture finally on how the pandemic actually works and how it spreads among the people here. We know that many other studies are on the way or have already been done in other parts of the world and the country, but I'm not aware of anyone which is so comprehensive, collecting so many different parts actually and different data from various areas. And finally, of course, the goal is to develop novel management strategies on the pandemics. What we apply here are technologies from all kinds. Actually, we use the normal PCR assays on, of course. We do it not by swabs, but by gargling and collecting the samples by gargling from the participants. We do next generation sequencing, not only for the virus genome, but also by single cell sequencing for T cells. for microbiome studies and so forth. We do, of course, serological studies. We do aerosol sampling and testing. We do evaluation of innovative test strategies. And here, this is maybe the most IP relevant area. We are kind of collecting from other institutes and other parts of the Fraunhofer Society the most innovative ways to either test infections and infection prevalence, or also looking into the immunological parameters and so on. So point of care tests or many other IP relevant or already IP'd technologies, we are trying to incorporate into this study in order to give all these partners actually the opportunity to evaluate and to maybe even validate their technologies with our large study here. And of course, we do a lot of modeling with the data. Finally, we have strong modeling groups involved here from Poland, from Germany, to really get the utmost possible out from this study. With respect to IP, as I mentioned, of course, IP might be interesting here for new approaches on SARS-CoV-2 research. We have IP, of course, from the Biomarker Center I presented, IP on prostate cancer, biomarkers on others, IP on, for example, also COPD markers and so forth. So that could be an interesting basis for collaboration in this context here. Yeah, that's actually what I have to say and to present in this short time. Thank you very much for your attention, and I'm really interested and looking forward to any potential collaboration. Thank you very much.
Thank you so much, Professor Dr. Friedman-Horn. I think you elaborated in your presentation about ways of collaboration, and there are so many ways of collaboration to discuss. I think we should, post this webinar, discuss also other ways of collaboration in respect to COVID and other perspectives as well. And I would like to call the next caller, the next speaker, sorry, Dr. Carmen Honor-Nugent. I'm sorry if I'm not pronouncing it well. He's from the Fraunhofer Institute of Toxicology and Experimental Medicine, ITEM, Integrated Platform for Liquid Biopsy-Based Personalized Cancer Medicine. Dr. Kameran, please, you are most welcome. I just would like to ask the other speakers if you can make it a bit short, because we are running out of time already. We are beyond schedule, so please, if you can just make it a bit shorter. Thank you so much.
Sure, thank you. So do you have my screen already?
Yes.
Okay, very good. So thank you very much for the kind introduction. So I'm representing the Fraunhofer ITEM, the Division of Personalized Tumor Therapy. We are located in Regensburg, Bavaria. So the core of our activities at the Fraunhofer in Regensburg is basically translational science with patient material, more in the context of oncology. However, we are also expanding to rare disease and recently also to COVID-19. To start with, I mean, as you can imagine, working with translational research with patient material, we would need to have a strong clinical network. So we have built a strong clinical network in the region from university hospitals from which patient tumor material is taken. So at the chair of experimental medicine at the University of Regensburg, headed by Christoph Klein, who is also the head of our Fraunhofer division, we basically have a diagnostic lab which is accredited, and the samples are basically processed there. On the one hand, these patient samples are used at the university for basic research, but we have also legal frameworks in place which allow the Fraunhofer group to have these material transferred from the diagnostic lab to Fraunhofer for basically different translational research. So the whole translational research is done at the university, at the Fraunhofer side, material coming from the university. And we have different groups at Fraunhofer. I'll come into in a bit, but the whole idea is that the constellation that we have here allows us to work with external partners, either being industry partners or coming from public sectors, from which we can basically receive R&D projects as a service provider, but as well, we can basically provide them our results in a cooperative manner. A problem which basically has to be understood in the cancer area is that typically in patients, once the primary tumor is identified, these primary tumors are removed through surgery. However, before the surgery, certain single tumors persist and they go to distant organs, initially form micrometastasis and later on full-blown metastasis. And the problem is that the molecular analysis is typically performed from that surgical tumor material and sort of extrapolated for later stages of the cancer, which may not necessarily be represented by the primary tumor. So there is a couple of years between the removal of primary tumor and the manifestation of metastasis. The problem, however, with metastasis is that due to difficulties and accessibility to metastatic material, it's very difficult to have biopsy material. So we are using liquid biopsy as a non-invasive system to basically have information about systemic cancer in the metastatic setting. So in this slide, you can see the whole structure of our division in Regensburg. So as mentioned, we have a strong clinical network and we receive patient material from different university hospitals through liquid biopsies. I mean, I'm in this talk focusing on liquid biopsy, but we also have solid biopsies. The first step, which is basically the group of Bernhard Polzer, is to identify and isolate single circulating tumor cells and also cell-free DNA. This sample preparation for cellular molecular diagnostic is the first step in the process. The cell-free DNA or single circulating tumor cells are either analyzed through NGS group, which basically has genomic and transcriptomic single cell sequencing abilities and biomarker discovery. On the other hand, it's also possible to generate preclinical models, either in vitro or in vivo, from these circulating tumor cells. Once these models are generated, we can run high throughput drug screens or target discovery projects. And basically all this information is translated back to the clinics for a therapy prediction so that the patients also receive the correct therapy based on this multi-omic data. So in terms of the single cell analysis, once these single cells are isolated, these single circulating tumor cells are isolated, we can either amplify the genome or transcriptome. And there are several different methodologies to do single cell genomic and transcriptomics, which have been established or are currently being established in our department. Many of these workflows have been automated and basically suitable for large scale samples. Maybe I will from here on focus more on the preclinical models. So I mentioned that we have access to blood in the case of liquid biopsy, but also cerebrospinal fluid in the case of metastatic patients from pleural effusions or ascites, but also primary tumors in the case of disseminated cancer cells from lymph node or bone marrow. Independent of the source of these tumor cells, we can either generate xenograft models from these rare cancer cells in vivo, or generate in vitro models either in 2D or 3D. The 3D systems can be either spheroids or organoids. Sometimes we basically generate models from in vivo and then transfer them into vitro if it's not possible to directly generate those models. Once those models have been generated, we basically characterize those models to make sure that those models are representing the original patient material. So what you see here is a profile of genomic alterations in the original circulating tumor cells from the patient, the xenograft model, and the patient-derived organoids. The red areas are chromosomal gains, and the blue areas are chromosomal losses. So what you can see from this slide is that there is a very good representation in the models, whether that is in vivo or in vitro, to the original circulating tumor cells that has been, in this case, derived from triple-negative breast cancer patients. So through these activities, we have generated a panel of different patient-derived preclinical models, currently a little bit more than 50 in the area of melanoma, lung cancer, either small cell or non-small cell lung carcinoma, breast cancer, pancreatic, and salivary gland. They either come from non-metastatic patients, so either primary tumors or lymph node, sometimes also bone marrows. In metastatic patients, we either derive these models from the blood or malignant diffusions, whether that is pleural diffusions or ascites. We have also expanded these activities to ovarian and bladder cancer, both in the area of adjuvant and metastatic settings. So once these models are being generated, the next step is to adapt them to high throughput screening. So here in Regensburg, we have a modern high throughput screening facility. What we do here is to use our drug libraries or siRNA libraries, which are in 384 well plates. These are transferred into assay plates, which are also 384 through acoustic dispensing using nanoliter dispensers or pipetting robot systems in the microliter range. Then the cells, the patient-derived models, are seeded in these assay plates. They're incubated. Then in terms of the readout, we basically have different readouts. These could be either homogeneous bulk readouts, for example, viability. We add viability readouts and measure the dose response of the compounds. Or we have automated fluorescence microscopy. So in this high content microscopy approach, we have the systems to do confocal high content microscopy and the follow-up image analysis, either by immunostaining or adding certain dyes to the cells, or in the case of functional readouts, we also have different systems to look at functional readouts. For example, calcium imaging. For example, we did a drug screen in cardiomyocytes, which is not in the context of oncology, but in those disease indications, we're also active. In terms of libraries, depending on the project, we use different libraries. We have cancer-specific libraries with either approved drugs or preclinical and investigational drugs. We have bioactive libraries, which are either the FDA-approved small molecules or drugs that are currently being investigated in clinical trials. Diversity libraries through our collaborations with Fraunhofer EMA. We have access to 200,000 small molecules if one is interested in high throughput screenings for novel drug discovery, and as well RNAi and CRISPR libraries for druggable genome and focused libraries. One application of these platforms is, for example, in the case of patients who are metastasized, either lung or breast cancer patients, or sometimes pancreatic patients. These generate typically these malignant diffusions. These are fluids full of tumor cells, which has to be taken out of the patients. In the case of pleural effusions and pericardial effusions, we have done several screens, which we initially isolate the tumor cells, characterize them, and enrich them. Similarly, we seed them in multi-well plates. We add our compound libraries, incubate them, and read basically the viability in the presence of drugs. So typically here we screen anti-cancer compound libraries, and so to say, predict a drug response to the tumor cells of the patients. And these drug sensitivities are basically transferred back to the clinics and used for patient treatment. And this is a very rapid process. It takes a week. Typically, we are faster than pathology in predicting drug responses. Importantly, in these contexts, we are not only isolating tumor cells, especially in these malignant fluids, it is rich of T cells. And in principle, it's also possible to look at immuno-oncology drugs and look at the efficacy of those in the autologous T cell cultures combined with the tumor, which can be either 2D or 3D organoids.
Dr. Kramer, I'm sorry, 2 minutes, please. Thank you.
This is my last slide, yeah. So in principle, I would like to summarize the platform that we have here. We have access to patient material, which comes from different sources. Through liquid biopsy, we isolate the single tumor cells or cell-free DNA that are biobank. Through NGS platform, we can predict drug responses to certain drugs for some of these patients. Through model generation and functional screening, we also can predict drug responses functionally. These are all going to bioinformatic analysis, and at the end, we try to have drug response prediction in most of the patients. So we are also open to different forms of collaboration. Either this could be a joint project or validation of certain systems with patient materials, or some of these technologies have been also patented, which we can discuss if there's an interest in that in more detail. Thank you very much.
Thank you so much, Dr. Cameron Holnar. I would like to welcome our last speaker, Dr. Engineer Andreas Taube. He's from the Automation and Digitization for Life Science Workflows, Department of Automation and Bioproduction, Fraunhofer Institute for Manufacturing and Engineering in Automation, IPM. Dr. Andreas Taube, please.
So thank you very much. You should hear me now. And thank you very much for the invitation today and to show you our...
Put your presentation, if possible, on viewers mode. Thank you.
I try to do this, yes.
Thank you.
So now it's better. Do you see it?
Yes.
Okay, so about automation and digitalization. So I'm from the engineering side of Fraunhofer. I am heading a group of nearly 30 scientists at Fraunhofer IPA, and we are also already working in the meaning of bioconvergence. So my team consists of engineers, but also of biologists and software developers, and we are developing software and automation solutions for life science labs. And from our opinion, the best solution brings these worlds together. It's robotics, it's software, it's the ease of use, and of course, it's the implemented protocol which should be work. And we had already collaborations with all the colleagues from our life science side at Fraunhofer, you heard before, and also to the industry or to industrial partners in the pharmaceutical industry, diagnostics, and other life science labs. So, you know, all life science innovation was accompanied by also technology innovations. So that's not new, but the possibilities that are coming up today, and the methods and also the different variants of technology you can apply to your process. This is quite very broad, and here I give you an impression. If I ask you how the scientific workplace of the future is look like, you see lab on a chip technology, you see this human-like robot, you see an assisted manual workplace, and you see also an integrated robotic plant which do everything completely automated, and also this hybrid version, mobile robotic system, which is also able to implement workflows in an automated way. So a lot of different possibilities, and our mission is to bring these worlds together to work on this technologies and help our customers and our project partners to manage this and to bring the best solutions for their individual process and their individual protocols into real application, and to do this fast and to do this sustainable, so that you can use these solutions also 5 years in the future. So therefore, we are looking for trends, for markets, and also for technologies which are already commercially existing. We develop processes along to an automated development. We integrate the end users in the lab by UCD and UX development. That's very important. There are people, and there will be people in the labs even in the future, and they have to use the technology. And so this integration of the end user is very important from our point of view. And yes, we develop technology. I will give you several examples on this. The lab automation, the connectivity of different solutions from different companies to bind this together, to do manual and process assistance, and to do software connectivity as well. So these are our working areas where we are working. So I hope that the videos are working. So the first example I want to show you is a modularized system. This is a system for automated cell culturing, and the special thing is what you see here. It is a system which is built up completely modularized, so you have the opportunity to build up these things, to implement very rapidly new components, new models. So this automation can be used as flexible as you are in a manual lab. Here you see a video of the building up of the whole system and the implementation of the process itself. So I have to hurry up a little bit, so I go a little bit forward. So here you can already see the process which is working, and how things are going on, and real processes can be adopted, and how this can be done. So another thing is the manual assistance. For this, we have also developed several solutions. On the one hand, it's a digital sample management system and an information system where we have digitalized the whole storage in labs. The main time waster in labs is searching for equipment. It's searching for the pipette, it's searching for materials and things like this. So if we digitalize this, this can be done easily. And you can also have a look on your processes. You give informations on the products, which consumables do you have used for a specific process? Does this has an influence on your process, and things like this? And the same can you imagine in the performance of the processes itself. I will show you a video after on so that you get an impression. So imagine a camera which is looking on your hand, and a monitor, a screen where you get informations about the working steps you have to do. That's what I mean with assistance, and that's what we have done here for normal lab pipetting workplace. An also important thing which we are working on is the application of augmented reality and also virtual reality, which is a very powerful tool to give the users in the labs an impression about or give informations about their complex working environment, and to give them the needed informations of the processes they are working on. So here you can see a simple example. These tools can give simple commands and the guidance for people through the labs. So this one you have seen already on one of the slides, mobile lab robots. It's a very impressive thing. Normally, if you want to use automation in a laboratory, you need space for it. So a mobile lab robot, it's able to use in a hybrid environment. It can collaborate with humans, and it can be used in a 24 hours way, but and it also can be linked to manual workplaces. So it's a hybrid thing how this can do. And so I'll show you a video. So the video is also too long, but here you can see Kevin in action. Of course, the video here is faster than it is in reality, but this is our lab here in Stuttgart. You see here a manual workplace at a sterile workbench. The robot is detecting the position of such a blade by detecting these position markers. And okay, here you can see the speed the robot is really moving on. The robot is recognizing the blade and is able to picking up this without teaching or anything like this. It's able to move through the laboratories. It's able to address now, for example, existing automation islands or standing alone solutions inside the lab, and that it is collaborative. I'll show you now. So this is the navigation of Kevin. You see there's a human and Kevin is driving around. It's detecting in real time what it is in its environment. Here you can see the people is working. This is the robot's movement, and that's how the robot is moving through the lab. Yes, it's a very flexible solution for a lot of different applications. So our vision is the connected lab, and yes, the connected lab is already reality. I think we are a little bit hard in time, so I skipped this. Please contact me if you are interested in this. And here I'll want to give you a brief overview of about some project results we are relatively proud about. We are concerning automated ATMP manufacturing. We are using AI for organoid handling and organoid sorting. We are concerning single cell handling. We are doing rapid integration. We develop customized automation solutions. We are doing robotics and vaccine production. So thank you very much. If you are interested in collaborations and in ideas of automate your workflows, then please contact me. Thank you.
Thank you so much, Andreas. And thank you all our distinguished guests and speakers. And thank you so much, Klaus, for participating with us in this webinar. And for all of our participants from Israel, thank you so much for being
Karina,
with us.
So I'm sorry to interrupt you, but Itai asked me to present the ideas for consortiums. Do you want me to present it now?
So
or?
I think we should have another discussion maybe with Fraunhofer separately. I'm not so sure it really relates to this webinar. But just to sum it up, we would send information to all of your participants via mail. with the information about the contact person in from our Fraunhofer side and the speakers, and how do you connect regarding their research areas and IPs and ways for collaboration, and information about our funding tool that was described by Aviv earlier, and any other information that you would like from us. Of course, we would send also the recording of this event so you can view it over. I think there's many ways to collaborate here with Fraunhofer as shown in areas of bioconvergence, for sure beyond that as well, but this is the focus of this discussion. So I'm really hopeful that we would see outcomes, positive outcomes out of this webinar. Please approach us if you have any questions, if we can help with anything regarding connections and additional information. Thank you all again, and have a wonderful afternoon.
Thank you very much for having us.
Thank you very much.
Thank you all.
Thanks a lot.
Thank
Bye-bye.
you.
Thank you. Bye-bye.
Thank you very much, Karina.